Hello, Vanakkam students. So here today we are back with this beautiful, awesome batch which is Dropper to Tropper. So today in this session we will be completing the most important chapter of class 11th, which is cell cycle. It is important in question wise also, and it is very much important if we talk about it in the concept wise. If you are able to understand mitosis, if you are able to understand meiosis, biology will be easier for you. So if you want to crack NEET today, this 2 hours is going to change your aspects for this chapter, which is cell cycle. So without wasting your time, let's quickly start this chapter. So today we will be discussing the chapter which is cell cycle. Now in my class I have some rules and regulations. Obviously if you're attending my class, I'll suggest you to do few of the things. What do you need to do? Sit with pen and paper. Okay, I would like everyone to sit with a pen and a paper. Whatever the concepts which I am going to tell you, write those concepts. If you are having any kinds of doubt in the live session, what you have to do is that just comment down. I will be checking the comments time to time so that I can see easily that which students are having doubts. Clear to everyone? Okay. Now let's start the session. Cell cycle, and today in this cell cycle most probably we are going to do with the mitosis. Now before that, let's have a general introduction. What introductions are? See, every organism on this earth, every organism, I repeat, every organism, you, small rat, to a big elephant, to a big giraffe, to a small amoeba, each and every one is made up of what? They are made up of cells. So can I write every organism is made up of cell? Yes sir, this is very true that every organism is made up of cell. You also agree with me? Yes. Now second thing is that if I talk about few of the things, so all the living organisms, almost, I should say, there are some exceptions, but almost all the living organisms do reproduction. So I can write this point also, that reproduction is done by almost all living organisms. Okay, now can you let me know that what are the living organisms, or which are the living organisms which do not reproduce? Anyone in the comment section. Now third thing is what? Each and every living organism, whether be it a small cell or be it a big cell, they all perform certain metabolic functions inside their cell, right? So if an organism is living, that particular organism is going to perform metabolic activities, and all this metabolic activities takes place inside the cell. Most accurately, I can also say that these metabolic activities are also done in cytoplasm, right? Inside the cell, cytoplasm is the main site where all these reactions are taking place. So this we had a basic introduction. Now what, sir? Now let us compare two organisms. One we have the unicellular and the other one we have the multicellular. Okay, so what is the difference between unicellular and multicellular organism? You will be like, sir, this we all know. Exactly, we all know the difference between the unicellular and multicellular organism, but do you know the difference in their cell division? That's the question. A class 9 student can also answer. Unicellular organisms are those organisms which are made up of single cell. Multicellular organisms are those organisms which are made up of more than 2 or 3 or many cells, right? This is a very basic definition and every one of us know. But if I talk about unicellular, let's suppose that, let's take our story, like there is an amoeba, okay, a cool amoeba is there, and this cool amoeba is having some structure in its center. So this structure is basically what? This structure is basically the nucleus. Let's have nucleus here. Okay. We have a nucleus. Yes, sir. We have a nucleus here. Now what? Now this amoeba wants to what? This amoeba wants to reproduce. This amoeba wants to reproduce. Now what will do this amoeba? How it is going to reproduce? Anyone in the comment section? I know you all know this, the reproduction. How does amoeba reproduce? Just by dividing it? Yes. So amoeba wants to do what? Amoeba, just focus this term. Amoeba wants to reproduce. Amoeba wants to reproduce, right? Yes. Now, during the reproduction, what happened is that they got like 2 amoebas are produced. Okay, 2 amoebas are produced. In the second scenario, this was the case 1. Okay, this was the case 1 in which amoeba wanted to reproduce. Amoeba wanted to reproduce. Now let's suppose we build a second case. Okay, in second case, amoeba said that I want to what? I want to grow. I want to become big. And amoeba was very happy that I will become big. But this amoeba, the amoeba which wanted to become big, when it started to grow, what happened? It again divided into 2 amoeba. And then the amoeba thought that this growth and reproduction is a scam. Yes, because for a unicellular organism, growth and reproduction means the same thing. Yes, amoeba wanted to grow, but it reproduced, right? So, for a unicellular organism, we can have this conclusion statement that growth and reproduction is same, right? But is this the thing similar to the multicellular organism? Is it similar to multicellular organism? Let's suppose that I got cut in my hand and some of my cells started to divide. And in the next morning, I am able to see another Harsh sitting beside me. Is it possible? No. So for multicellular organism, growth and reproduction are very, very, very different terms. So I can write here growth and reproduction are different terms. They are very different. Yes or no? Yes, sir. Growth and reproduction are very different in terms of multicellular organism, and in terms of unicellular organism, it is kind of similar. We got a conclusive statement. Now, each and every organism is made up of cell, and growth is basically what? Due to cell division. Reproduction is basically due to what? Cell division. If I talk about the multicellular organism, our germinal epithelial cells divides to form gametes, and then that gametes fuses to form zygote, and then we are produced, right? So everything, whether it's a multicellular organism or whether it's a unicellular organism, undergoes cell cycle. And why cell cycle, sir? What is the need for this cell cycle? The need for the cell cycle is what? Because this is a process through which a cell is dividing itself. Okay, so now let us understand the definition of this cell cycle, like how you are going to understand this. So it is what? It is a very genetically controlled process. It's not like that, okay, let's divide. So anything is happening at any time. If we want to divide genes, okay, let's divide. After that, what should we divide? What should we do? Let's divide mitochondria. No, it's a very, very controlled process. So who is controlling? Obviously genes are controlling. Yes. So can I write here genetically controlled? Genetically controlled. In today's video, I'm going to give you such a beautiful concepts about the chromosomes and all that you're gonna love it. And there will be no doubt, not a single point of doubt about in understanding what is 2n, what is n, where C, where 4C, where 2C. No doubts. There will be no doubts. I guarantee that. Now, this is what? This is genetically controlled. Okay. Next is what? What happens basically, sir? Like I understood, like cell gets divided in cell cycle, but what's the main thing here? See, the main thing behind the cell division is to divide the genes, right? So here in this, what happens? Number one thing, DNA replication takes place. Yes, obviously it has to take place. DNA replication takes place. Without DNA replication, if suppose that I'm gonna produce the baby and my DNA says that no, I'm not gonna go in the second cell, then how my genes or how my characters will be inherited to my babies, right? So DNA replication is the key factor for the inheritance. So this will happen in cell cycle. Second thing is what? Second thing here is cell growth. Yes, obviously, sir. Without cell growth, nothing can be possible. So if I say about the cell cycle, so cell cycle doesn't mean that reproduce. No. Cell cycle is basically what? In this cell cycle, DNA replication is also taking place and cell is growing normally as well. That means reproduction and other basic things are also going in the cell cycle. Now, after doing this, like DNA replication, cell growth and everything, at the end of the cell cycle, we get two daughter cells. So at the end of this cell cycle, what do we get? What do we get? End of this cell cycle, we get what? Daughter cells, exactly. We get daughter cells. Now, should I write here 2 daughter cells or 4 daughter cells? No, let's play a very safe thing here. Let's just write daughter cells. Why? Because if I'm talking about the mitosis, generally 2. In meiosis, at the end of meiosis, we get 4. So it's better to avoid confusion. Let's write daughter cells only. Okay. Now, let's move forward and let us have some more things, which is time duration. Sir, I am very interested to know that how much time takes place to divide a cell. You might have heard in somewhere like bacteria divides in 20 minutes. Yes, it's true. So let's have some names of some organisms, and I'm not writing this for fun, okay? This is given in NCERT and you have to remember it. So number 1 organism, let's start with the human beings. We are human beings. We are a super duper species on this earth. So let's give priority to ourselves. So human beings is what? Human beings. Sorry, I should say human cells, not human beings. How can human beings divide, right? So human cells takes approximately 24 hours to divide, okay? 24 hours to divide, and you will be like, oh wow, it's a such less time. But no, you are in a very great dilemma, because if I talk about this yeast, so this yeast is what? It's only taking 90 minutes. It's only taking 90 minutes to divide. While you prepare for NEET 2024, just imagine how many yeasts are produced and how many yeasts will die and complete their life cycle. Next, we are having bacteria. So as I mentioned, bacteria generally takes 20 minutes to divide. Okay. Everything has to be learned. This is not for fun. Everything, all the data must be in your brain. Let's come quickly revise. Human cell, 24 hours. Yeast, 90 minutes. Bacteria, 20 minutes. Very good. Now sir, why sir, why the cell needs division? Life was going very well. The amoeba was crawling. The amoeba was doing phagocytosis with the help of pseudopodia. Then what was the need for amoeba to divide? What was the need for our cell to divide? So for the division, for the unicellular organism, I could say that it leads to reproduction, right? So to maintain the species, obviously, but let's not focus on that reason. Let's focus on the cellular reason why the cell wants to divide. So number 1 thing is that let us draw a cell here. Okay, inside the cell, what we are going to have? Inside the cell, we are going to have a nucleus. Yes sir, we are definitely going to have a nucleus. Okay, now I'm going to give an example. Let's have a kid who is very naughty, okay? And this kid, when present in front of the father. Okay, this is his father. Let's suppose this is the father and this is the kid, or I should say this is what, this is a naughty kid, right? Naughty kid. So in presence of the father, this kid will be like, no, I am a very good kid, sir. I don't do anything. I read, I wake up on time. I'm a very good kid. But as soon as this kid is removed into another room, this kid will start dancing. This kid will get out of control, right? So same thing happens with this nucleus also. You all know that nucleus is what? It is a brain of our cell. Exactly, sir. So how can we relate this kid and father example? Let's see here. So let's suppose a cell got bigger and nucleus is now not able to control the whole cell, and this is the time when cell, or I should say nucleus, decides that we need to divide. Okay, now how can we understand this with the help of terms? So first of all, I am going to introduce a term which is called as karyoplasmic index. Okay, which is called as karyoplasmic index. I'm not writing the full form, you just note it down. Karyoplasmic. Okay, what is this? So first of all, we have a small cell. This one was small cell, and this small cell. Hi Akash Yadav, if you have a doubt, please let me know in the comment section. If you have a doubt, please definitely let me know in the comment section. Now, if I have this Ki, which is karyoplasmic index, then, this was a small cell. Okay, this small cell grew and made a big cell. Okay, made a big cell. Now what, sir? Karyoplasmic index ratio is basically what? It is basically the, for a large cell, if I talk about, it is very, very low. It is very, very low. And for the small cell, it is high. Okay. How it is calculated? What is karyoplasmic index ratio, sir? So see, I'm going to write a formula for you. Volume of nucleus divided by what? Divided by volume of cytoplasm minus volume of nucleus. This is what? This is the karyoplasmic index ratios formula. Okay. Now, what is happening if karyoplasmic index is low? If karyoplasmic index is low, that means what? This volume of cytoplasm is high. Yes or no? If karyoplasmic ratio is low, then the volume of cytoplasm is high. I hope that it's visible to you, right? If karyoplasmic index is low, then the volume of cytoplasm is high. And if the volume of cytoplasm is high, nucleus will not be able to control the whole cell, right? So for the big cell, what should we do? For the big cell, we can understand. I'll take the another slide so that I can make you understand better here. Okay, see, so for a big cell, I could say that karyoplasmic index is what? It is high. For a big cell, the karyoplasmic index ratio is what? High or low? It's low. Why, sir? Why it is low? Because here cytoplasm is more. Here cytoplasm is more, right? And for a small cell, the karyoplasmic index ratio will be high. Why, sir? Because cytoplasm as compared to nucleus is less, right? And just now we discussed a formula. Which was what? Which was volume of nucleus divided by volume of cytoplasm minus volume of nucleus. Yes or no? And if the volume of cytoplasm is what? If the volume of cytoplasm is high, then this will be high, and so karyoplasmic index ratio will be low. And that's the reason why cell will divide. And if I talk about the small cell, so for the small cell, this ratio, this denominator will be what? This denominator will be high. So for the small cell, the denominator will be what? The denominator will be low. and this will make what? This will make high karyoplasmic index. So very to the point that karyoplasmic index ratio is what? It is high for the small cell and low for the big cell. When the karyoplasmic index ratio goes low, cell takes a decision and divides. So this is the reason why cell is dividing. Now, let's move on and let's talk about the phases of cell cycle. So we have discussed like what is cell, why there is a need for division, what is the difference between the growth and the reproduction thing, everything we have discussed. Now let's enter to the cell cycle, main cell cycle. So in the main cell cycle, we have what? We have phases. So how many phases are present, sir? See, we are going to discuss everything here. So now we have what? And basically we will be dividing this cell cycle into two parts. Okay. One is the interphase. One is the interphase. And the second one will be what? The second one will be anyone. The second one will be M phase. Okay. So we divided the cell cycle into two parts. One is interphase and the second one is the M phase. Okay, great, sir. Now, what happens in the interphase? So interphase is basically what? Interphase is a preparatory phase. Interphase is what? Interphase is a preparatory phase. Let's suppose that tomorrow is your exam and you are preparing, or let's take a better example that we have NEET 2024 on 24th May. Sorry, 5th May, what I'm saying. NEET 2024 on 5th May. Okay. But you have started preparation right now. So this time for you is the interphase. That means all the preparations, whatever the preparations a cell need for division, they are going to do in the interphase. Right. And M phase is the D day. Like your NEET day is your M phase, in which the actual cell will divide, right? So in interphase, this is clear that in interphase there will be no cell division. In interphase there will be no cell division, but in M phase there will be the actual cell division. So sir, what's the need of interphase? We could have directly gone into M phase, right? No, it can't happen because it needs some preparation. So in interphase, what things happen? We call it as a preparatory phase. We call it as what? Preparatory phase. Okay, done, sir. Next, what? Next we have cell growth takes place in interphase. Cell is growing also in size. Okay, so normal cell growth takes place in interphase. Cell growth is taking place. Now what, sir? Now the cell is metabolically very active. Metabolically means what? It might be making proteins, it might be doing all the metabolic activities, catabolism, anabolism, everything the cell is doing in this interphase. So can you write here? Metabolically active, very active. Okay. Next, what do we have? We have, this is what? This is the longest phase of the cell cycle. Yes. This is what? This is the longest phase. So let's write here longest phase. Now my question is here that why this is the longest phase? Why this phase is known as the longest phase? Anyone? Because preparation needs time, right? In NEET, it's exam for 3 hours, 30 minutes, right? But for preparation, we need years of hard work, right? So for interphase, it's preparing things. It takes lots and lots of time. And then M phase takes very small time, okay? Now, in the interphase, we have again what? In the interphase, we have again few of the phases. So what phases we have? Let me show you once. Yes. So in interphase, again we have G1 phase, G2S phase and G2 phase. Interphase is again divided into G1 phase, S phase and G2 phase. Yes or no? Okay. So cell cycle is divided into interphase and M phase. Now Interphase is what? Interphase is again divided into G1 phase, S phase and G2 phase. Clear? Now I'll just go give you a very brief idea of this G1, S and G2 phase, that what happens in this G1, S and G2. Okay. So in G1 what happens? In G1 DNA replication gets initiated. I didn't said that DNA duplicate. No. DNA duplication or replication is initiated here. So let's write here DNA replication initiated. Right? Now what happens? You have to remember this, that in this condition no DNA is replicated. It's just got started. Okay. Then we have S phase. S phase is known as synthesis phase. And in this phase, actual DNA replication takes place. In this phase, DNA replication occurs. DNA replication occurs in this thing. Clear to everyone? So I told you that in G1, it starts. In S, DNA replication occurs. This is a basic idea I am giving. I'm not talking about that this is the only thing which is going to happen. We are going to see each and everything in very detail. But this is the basic idea. Let's build a basic thing. Let's move step by step. Okay. G1, DNA replication. G1, DNA replication started. S, DNA replication occurred. What about G2, sir? What about G2? Where should I write? I'll write G2 here. Okay. So what about G2? So in G2, if I talk about in G2, whatever proteins are needed for the M phase, those proteins are made. Okay. I'm gonna write here proteins for M phase. And the important part is that during the G2 phase, they are making tubulin protein. They are making what? They are making tubulin protein. Now, there is one more thing, a very important protein, very important protein, which is what? Which is the histone protein, a very important protein, which is known as histone protein. So I would like to ask, what is the work of histone protein? What is the work of histone protein? Then you will going to say, sir, that histone protein is that protein which helps in what? Which helps in packaging of the DNA, right? They helps in the packaging of DNA. Clear? So if they are helping in the packaging of DNA, then whether it should be made in G2 phase or S phase, obviously DNA replication is taking place in S phase, so packaging might be needed in that phase only. So histone protein is the protein which is what? Which is formed in S phase. So let's write here histone protein. Where it is formed? Histone protein is formed in S phase. Clear to everyone? I hope that this thing is pretty much clear. And this is what cell cycle is. Now we have discussed about the basic things, and when we are going to go in very detail, it will be very easy for you to understand. Now let us go with the M phase or mitotic phase. Okay, so in M phase what happens? You will be amazed to know that this M phase just takes what? Just takes 3% of the time. Like 97% we are doing what? We are doing the preparation. 97% we are having this thing, preparation. And 3%, just the 3% we are doing what? We are doing the actual work in the M phase. Now I would like to ask you a very good question. In M phase, how many phases are there, sir? So they are prophase, metaphase, anaphase and telophase. Just 4. We just need to discuss this, and then this session will be completely over, and you will be able to solve each and every question of this particular section. Now let's move on and let us discuss about the mitosis, right? Which is M phase basically. Okay, so M phase is what? In M phase, mitosis is going takes place. Here what happens? What happens, sir, in mitosis? So in mitosis we have what? We have the equational division, sir. Mitosis is what? It is an equational division. Why it is an equational division? How can we say that this is an equational division? Because see, if I am a person who is having 46 chromosomes, okay, if I'll divide through mitosis, I'll again have 46 chromosomes. That means chromosome number doesn't get changed in mitosis. So I can say it is equational division. And that is why it is what? It is equational division. Clear? Okay, sir. Now, what is the property of mitosis? The second property of the mitosis is that it takes place in somatic cells. Now, sir, what is somatic cells? The cells of your body which do not take part in reproduction, like your hand, like your hair cells, like your cheek cells, like your ears. So all these cells are what? All these cells are somatic cells, and in this cells mitosis takes place. Now second is what? Third point. Let's have the third point. I told you that 2n will be 2n if suppose a body is what? A body is diploid. So after the division it will be diploid. This is clear. Next, this mitosis is generally what? It is generally seen in the somatic cells. Now there is one thing, when we have an exception here. Exception is what? Exception is that in some haploid cells, like in drones, mitosis takes place. In drones, mitosis takes place. Drones are what? Drones are haploid. Now, next we have in plants. In plants, if it is haploid or diploid, in both condition, mitosis will only takes place. Mitosis will only takes place. If it is haploid and diploid, whether it is a plant is haploid or a diploid structure, mitosis will takes place. But in animals, in generally, it occurs in somatic cells, which is a diploid structure. It doesn't occur in the formation of the haploid structure, right? If we need to form the haploid cells, mitosis can't work. Clear to everyone? Now, let's move on and let us discuss about some few concepts. Now, from here, the main cell cycle is gonna start, and I want everyone to pay very much attention here. Okay, let's take 2%. Let us take 2%. So here let's have this person. Okay, let's name them. Let's make a character. Whenever we are going to take an example in biology, we'll call them. Okay, so let's name it Harry. Let's name it Harry, and let's name this girl as what? Any name. Let's name this girl as Julie. Okay, so we have Harry and Julie, two characters of our batch. Okay, what do we have? We have two characters of our batch, which is Harry and Julie. So whenever we are going to take example, let's have Harry and Julie. So this Harry is what? This Harry is male and the Julie is female. Now, they both are human beings. Yes, sir, they both are human beings. So Harry will be what? It will be diploid. Yes, it will be diploid. Julie will be what? Julie will also be diploid. Right? Both of them will be diploid. Clear? Now, both of them wants to produce baby. Okay, both of them wants to produce baby. So here we have gametes. To produce baby for reproduction, they have to make gametes. Yes or no? Obviously. If they are making gametes, then in gametes, what is going to happen? In gametes, we are going to get half of the chromosomes, right? In gametes, while making the gametes, we are going to get half of the chromosomes. Okay, sir, that's great, sir. Now, if suppose that this one is one of the chromosome, this one is one of the chromosome here, and this is Julie's chromosome. See, I'm making you understand a very good concept. No one on the YouTube is going to explain this concept better than me. I bet that. Now see, this is what? This is a chromosome, and in this chromosome, there is a gene which is controlling your hair color. Okay? In this chromosome, there is a gene which is controlling your hair color. And again, in Julie's chromosome also, there is one gene which is controlling hair color. Okay. Now, when these gametes will be fused, when this gamete will be fused, so we have this while fusion of gametes. So gametes were what? Gametes for N, and Julie's gamete were also Julie's chromosome or gamete will be N. When these gametes is gonna fuse, you will have what? You will have 2N, which is a zygote. Yes, sir. Very clear. Now, let us understand about the chromosomes. What happened to the chromosome? So, this was the chromosome of Harry, and this was the chromosome of Julie. Now understood from where and how we have got two chromosomes. One chromosome came from our mother. One chromosome came from our mother, and the other chromosome came from our father. Okay, I made Harry female. 1 second, I'll write here father. Okay, otherwise Harry will be like, what is this behavior, sir? So let's make him as father and let's make her as mother. Okay, so this is how we are gaining the chromosomes from our parents. Clear? Okay, sir, this is very much clear. Now this was N. Harry made a gamete which was N. Obviously this was N, and Julie made a gamete which was N. But we are what? We took both the chromosomes from our parents and we became 2N. And what? And we became 2N. I hope that this is clear. Is it visible to everyone? Okay, I'll be little side enough so that you can see the screen. Okay, now let me make myself little small so that you are able to see the screen very clearly. Okay, now it's fine. Now let's get back to our topic. So we discussed father, we discussed mother, and we saw that how NN is becoming 2N. Now this was very easy. We have read in class 10th also. Sir, say something, explain something more good. Okay, I'm doing that. Wait for a while. So here, after this thing, we are going to understand some basic concepts which you really want to know, and this will be very, very informative. And after this video, after this thing, you will never be confused about what is 2C, what is C, what is N, what is 4N. Everything will be cleared. So let's discuss some important concept here. Okay, I'm making that diagram again. So this was Harry's chromosome. This was Harry chromosome. That means the father, the male father chromosomes. And this was N. Yes, sir, this was N. We got it. Now what? This was the Julie's chromosome, which was what? Which was again N. Yes, sir, we got it. Okay. Now, combining both of them, we are formed, like from our father we got N, from mother we got N, and we formed 2N. Okay, sir, that's very good. So this is what? This is 2N. And this is known as homologous chromosome. If suppose that this is the chromosome number 1 which you got from your father, and this is the chromosome number 1 which you got from your mother, and both the chromosome sitting together inside your body, this makes the homologous chromosomes. Now I hope that this concept of homologous chromosome is very much clear to you. This is what? This is the homologous chromosome. This is we are going to call as homologous chromosomes. Is it clear to everyone? Yes or no? I hope it's clear. If you have doubts, let me know in the comment section. Now, this is clear, sir. Now, again, the thing is that there are questions like what is chromatids, what is chromosomes, how many chromatids, how many chromosomes. So everything I'm going to discuss here. Wait for a while. Each and everything. This is a batch for dropper to topper, right? That doesn't mean that I'm going to go very fast and I'm not going to explain basic things. We are going to do from basics, and we are going to prove that droppers can be a topper, right? Okay, now see this chromosome. Okay, can you see this chromosome, this white one? Yes sir, it's visible. Okay, so if suppose I'm talking about, we have one chromosome. Okay, one chromosome, any one chromosome, take any one chromosome, whether of Harry or Julie. Okay, so if this is Harry and Julie chromosome, so can I say that this thing, this structure is what? This structure is known as chromatids. These are what? These are chromatids. Okay, these are chromatids. Okay, in a similar manner, in Julie's chromosome also, we have what? We have this chromatids. Yes sir, we have this chromatids. Okay sir, so can I say in one chromatid, one chromosome, how many chromatids? In one chromosome sir, one chromatids. One chromosome, how many chromatids? One chromatids. If I'm talking about N, if I'm talking about the haploid thing, so a haploid chromosome, one chromosome is having one chromatid. And if I talk about how many of this 2N, how many this 2N contains how many chromatids? So you are going to say in total 2 chromatids. Yes or no? Now is it clear? Is it clear to everyone? Now this way only we are going to understand about what is 2C, C and 4C. Okay, so we got it. One single chromosome, one chromatids. 2N, 2 chromatids, right? Clear? Now what about this thing? What should we call this 2 chromosomes? So these 2 chromosomes are what? Are the sister chromosomes? Can we say them as a sister chromatids? No, sister chromatids will not be used here. They both are what? They both are homologous chromosomes. And what is sister chromatids? We are going to see in the next few minutes. Okay. Now, is it clear? One chromosome, one chromatids. Again say one chromosome, one chromatids. We have N, one chromosomes, 2N, 2 chromatids. Clear? Now, let's move on. Let's move on. I'm gonna ask you one question. If suppose I'm having 46 chromosomes, okay? Everyone in the comment section, I need the answer. If suppose I'm having 46 chromosomes, clear? Then what will be the number of chromatids inside me? What will be the number of chromatids? Fast. 46 chromosomes, how many numbers of chromatids do we have? Sir, the answer to this question is 46 chromatids. Yes, the answer to this question is 46 chromatids. Okay, how sir? We discussed one chromosome, one chromatid. If I'm asking about N, so what sir? So you are telling about the N. N means one chromosomes, right? So 46 chromosomes, 46 chromatids. But if suppose I have asked you that 23 pairs of chromosomes, 23 pairs of chromosomes, that means I am talking with respect to the diploid organisms, then you would say that pairs means what? One pair having 2 chromosomes. So 23 into 2 chromatids will be our answer. So can you see this? How we can understand here? If they are talking about the pairs, multiplied by with 2. If they are talking about the just chromosomes, so whatever the number of chromosomes is given, just answer that. That will be your answer. The number of chromosomes, simply write the answer. Pairs multiplied by 2. Clear? Okay, sir. Now, let's again move. Let's get back to the S phase. Let's come towards the S phase. S phase. Okay. So what happens in S phase? What happens in S phase, sir? So see, during this thing, during S phase, we have seen that okay, chromosomes get divided in S phase. So something can happen, and we will just read it out, and we will mug up the things, and we will leave the concepts. No, if you are going this cell cycle, you can't solve questions if the questions are twisted, which is twisted in the NEET, so you can't answer those questions very easily. So let us understand what is S phase, okay? Like what's actually happening in S phase. So see, before S phase, okay? Before S phase or before replicating, we had Harry, okay? We had Harry and we had Julie, okay? We had Harry and we had Julie. They're chilling, nothing, no reproduction, the cells are chilling in their body. But suddenly what happened? Volume of cytoplasm increased, and then the nucleus was like, gave the command for cell division. Now what is going to happen? In S phase, these chromatids are gonna divide. How, sir? See, this is the Harry's chromosome. So it will make a copy of itself, okay? It will make a copy of itself. Harry's chromosomes made a copy, replicated itself. And again, let's have this Julie's chromosome. So Julie's chromosome also in the S phase replicated. Clear? Okay, so this is very much clear. So this was what? This was the before S phase and this was during S phase. During S phase. Okay, now what happened? What is going to happen here? See it. Just observe it very carefully. This is very, very easy to understand. We discussed that this was what? This was N. Okay, sir. This was N. Okay, sir. And this whole made what? This whole made 2N. Yes. Okay, clear? Clear. Now what? Now what, sir? This is N. This is N. This is N. This is N. Okay. Can I say that this is one chromosome and this is also one chromosome? Is it visible? Is it visible? Yes, this is one chromosome. The chromosomes are just duplicated. They are not torn apart from each other. They are still together. So this is what? The whole thing is what? Again, this is N and this is N. We can't say that it became 2N. No. Each of the let's suppose this was the chromosome number 1 from father, right? And this was chromosome number 1 from mother. Yes or no? Still, they are what? They are N and N. And this N and N is going to make what? It is going to make 2N. So, is there any change in the ploidy? Is there any change in the ploidy? We have what? We had 2N before also and 2N after also. No change in ploidy, guys. No change. Why? Because this is the single chromosome. Why you are seeing this chromosome as 2? No. It is a single chromosome. It's still a single. See? It's a single chromosome. It is also a single chromosome. Then the chromatids are not apart from each other. Until the chromatids are not apart, we are not going to consider it as a different chromosome. So both the chromosomes are still single. So we are going to call it as N and N. And both of them is present inside the same body. So we are going to call it as 2N. Right? N and makes 2N. That's very simple. Right? Clear, sir? This much is clear. Okay. Now, these were what? They both are what? They both are still homologous chromosomes. Right? Now, let's move on. And now let's talk about the number of chromatids in this case. Number of chromatids in this case. Okay, so number of chromatids in this case. What happens here? See, earlier what happened was one chromosome, one chromatid. Earlier we were discussing one chromosome, one chromatid. Okay. But now what happened? In S phase, magic happened, and magic not happened. Basically, what happened? DNA duplicated, right? So one chromosome is now having 2 chromatids. One chromosome is now having how much chromatids? 2 chromatids. Is it clear now? Yes or no? Clear? Yes, sir. Very much clear. Now see. Now if I ask you that if suppose I have 46 chromosomes, if suppose I have 46 chromosomes and this 46 chromosome is present in the S phase, I'm talking about the 46 chromosomes of S phase, then how many chromatids do we have? Then how many chromatids do we have? Anyone in the comment section? How many chromatids do we have? Yes, apply your brain. Normal condition, in normal condition, what did we have? We have 1 chromosomes, 1 chromatid. Now the cell is in S phase, so I'm talking about 46 chromosomes is having how many chromatids? So sir, in S phase you told that 1 chromosome is having 2 chromatids. So 46 chromosome will be having what? 46 chromosome will be having 46 into 2 chromatids. That's very good. We will be having 92 chromatids. How many chromatids we will be having? 92 chromatids. So can you see that? Before the S phase, we had only what? 46 chromosomes, 46 chromatids, right? And now what did we have? 92 chromatids. So can I say that DNA got doubled, the content got doubled? This is how it is what? From 2C to it became 4C. Earlier that DNA was what? Earlier that DNA was 2C and now it's 4C. Clear to everyone? This is the concept behind 2C and 4C. I'll repeat again. Okay, I will repeat again. Don't worry. So earlier what we saw? Earlier we saw that this was what? This was 1C. This single chromosome was 1C and this was 1C. Before S phase, we had N and N. Each chromosome was having 1 chromatid. So in 2N diploid organism, we had 2 chromosomes. So it was 2C, right? C. So this was the earlier condition, yes or no? Yes, sir. This was N, this was N. This is having this was 1 chromatid and this was 1 chromatid. And since it's a diploid organism, so this will have in total 2C content. Yes, sir. Now during S phase, what happened, sir? During S phase, they both got divided, right? So we had this and we had this, like both the chromosomes replicated itself. And now what is happening? That both the chromosome, this chromosome is having 2C and this chromosome is having 2C and both of them will make 4C. And this is what 4C means exactly. This is what 4C is. So now I hope that the concept behind this is very much clear to you. If it is, let me know in the comment section. So I can say that the DNA content is what? The DNA content is doubled. Now I'll ask you some questions. Okay, can I ask the questions? Yes, sir, definitely. By the way, don't have an option to say no. Okay. Now we have N, we have 2N, we have 4N. Okay. N, 2N and 4N. Okay, sir. We have what? We have N, we have 2N and 4N. So in N, sir, what? In N, how much was the DNA content? Just see about the N. In N, we have 1C. If a single chromosome, how many content? How many chromatids? C. If 2N, 2C. If 4N, then it will be 4C. Right? Clear? Is that so? 4N means what? 4N means 92 chromatids. Okay. We are talking about the number of chromatids here. Okay. If suppose the cell is haploid, how many C? Just 1C. If we are talking about the diploid, 2C. And in the similar manner, if I'm talking about the 4N, it will be 4C, right? It's clear. I hope that this concept is very much clear to you all. Now, let's move on and let us discuss about the chromosome structure. A very basic discussion of the chromosome structure will be there so that we can use that information in cell division. Structure of chromosome. Structure of chromosome. Okay, sir. So what is the structure of chromosome? So in the between we have what? We have a circular disc-like structure and in this only the chromatids are attached. Okay. I'm talking about the chromosome which is present in the S phase, okay? I'm talking about the chromosome which is present in the S phase. Clear, sir? Now, this structure is known as what? This round structure is known as what? This is known as centromere. This is known as centromere. So let's write here centromere. Okay. Now we have these two chromatids. Yes. This is what? This is a 2n. This chromosome is basically what? Is it 2n? Is it 2n? No, this is n. This is n only, but this chromosome is of S phase, and that's why it is having two chromatids, right? So these both chromatids now are known as sister chromatids. That means if the chromatids are attached to the same centromere, it is known as sister chromatids. Clear? Same centromere, sister chromatids. I hope that this thing is clear to you. Now, what is kinetochore, sir? We have always heard about the term kinetochore. So let's see what is kinetochore. Here we have some structure, and this structure is known as kinetochore. So what does this kinetochore helps? This kinetochore is basically a place where spindle fiber is gonna attach, and it is gonna divide the chromosome into two parts, right? So kinetochore basically what? Basically helps in attaching of spindle fiber. Helps in attaching of spindle fiber. This is the basic work of the kinetochore. And if you see this kinetochore, so it is basically a disc-shaped structure. It is what? It is a disc-shaped structure. Clear to everyone? Okay, sir. Now this much is clear. Let's move on. Now, let's quickly revise the concept. Chromosome before S phase and after S phase. Okay. So this was what? This was the 2C content. Was it? Yes, obviously. This was what? This was 2C. When? Before S phase, and this was 2n condition, right? Yes, sir. Why 2n? You can see here it is n and here it is n. How many chromatids are? 2 chromatids. So that's why 2C. We have 2n and 2C condition before S phase. After S phase, what happened? After S phase, what happened, sir? Wait, I'm gonna draw it. So after S phase, this chromatid got replicated, right? In a single chromosomes, we have the replication, and this is how they became C to 2C. They were C, right? To 2C, and this 2C 2C made 4C, but still they are N and N because chromatids are not yet separated. So it is 2N. This makes every concept clear. Now let's move on and let us discuss about some of the main things. Let us now enter into the M phase. So I'll just revise quickly what things we have discussed. We discussed about what was, how N is formed, how 2N is formed, what is homologous chromosomes, how C, what is actually C, what is 2C, how 2C becomes 4C in S phase. Everything we have discussed. Now let's enter into the mitosis, and this is a very easy thing. We have discussed the concept now. Now it will be very easy. If you have missed any part of this video, after this live class, watch this video. It is gonna change your perception of on how the cell cycle chapter you have to study. The very great concepts we have discussed today. Now let's talk about the mitosis. So in mitosis, how many phases are there, sir? We have 1, 2, 3, 4 phases, and which is the very easiest part now. We have the prophase. Then what we have, sir? We have metaphase. And then what we have? Anaphase. And then we have telophase. Clear? How many phases? Prophase, metaphase, anaphase, and we have this telophase. Clear? Okay. Now, what happens in prophase? See, I'm going to make you understand just with the help of diagram only. Just the diagrams will be beneficial for us, and after that we are going to write the theoretical part. Okay, so let's go on with the prophase. Prophase. So in prophase, it is divided into two parts. One is the early prophase, early and late. So in early, what happens? Let's have a cell here, okay, and inside this cell we will be having a nucleus. Okay sir, we will have a nucleus, and then what? We are going to have DNA, chromatin network and everything, right? And then we have what? Then we have this two structure, which is what? Which is centrosomes. Exactly. So they, in prophase, these centrosomes starts moving where? They start moving towards the poles. Okay, so I should write here, number one thing which happens in the prophase is centrosomes where they are going starts moving towards pole. Clear? Yes sir, it's clear. Now what happens in late prophase, sir? What happens in the late prophase? Then we will going to discuss, but before that there are some other things which happens in early prophase. So here condensation starts in early prophase. Let's discuss early here only. Let's discuss the late in the next page. So during this, the chromatin network starts condensation. So can I write here, condensation gets started. Condensation is started here. Okay sir, that's very good. Now what sir, what happens again? The chromosomes were tangled. They are just like they are very much tangled. They are just mixed up, just like the wires here. They were so much tangled. Okay, but now what? They are getting untangled. So I can say that the chromosomes are getting untangled here. Okay, great sir. And prophase comes after what? Prophase comes after the interphase. So can I write here that it follows interphase? It follows interphase, which means interphase is before prophase and prophase is after interphase, right? So these are the things which happens in the prophase, or more specifically early prophase. Now let's move on with the late prophase. Okay, so in late prophase what happens, sir? In late prophase, mitotic chromosomes are formed. The chromosomes which are now called as what? Mitotic chromosomes. And please make it a star mark. From here the questions can come. Mitotic chromosomes is found in, so mitotic chromosomes are found in late prophase. Now here what is going to happen? You are going to see that there will be what? There will be two chromatids and with one centromere. What things we will having? Two chromatids. Chromosomes will have what kind of structure? Two chromatids with one centromere. With one centromere. This will be what? This will be the kind of structure in the prophase 1. See, here we will be having, and this. Why sir? Why this is happening? Because it is after the S phase, DNA has duplicated, and that's why we are having two chromatids and one centromere. Two chromatids and one centromere. Okay, clear? So this will be the structure of chromosomes we are going to have in late prophase. What else, sir? What else? Here all the cell organelles, whatever is not required for during the cell division, they all are disappeared. So cell organelles gets disappeared. Cell organelles gets disappeared. Clear? Okay sir, this is much clear. Now Nuclear membrane also starts to degenerate. They also starts to degenerate. Why, sir? For division, the chromosome has to come out. It needs space. It is going to be condensed. The centromere is going to generate spindle fiber. And if nuclear membrane is present, how they are going to divide, right? So nuclear membrane also gets what? Also gets disappeared. This much is clear to you all? Okay, sir. Now what? We discussed mitotic chromosomes is formed where? Late prophase star market. Okay. 2 chromatids, 1 centromere. This is clear. Cell organelles gets disappeared. That's very good. Nuclear membrane gets disappeared. This is also very good. So all the steps we have discussed in the late prophase. Now what happens, sir? I can say that this prophase is followed by what? It is followed by the M phase or telophase or anaphase or metaphase. What? See, if I am saying that I am being followed by, that means which thing is what? Which thing was before me? That was G1 or G2. We had G2, S and G1, S and G2, right? So we will be what? We will be saying that if suppose I am saying that G1 phase is followed by what? G1 phase is followed by M phase. That means after G1 what came? M phase. If I'm going to say that S phase is followed by what? S phase is followed by G2 phase. So I can say that prophase is followed by what? Prophase is followed by metaphase. Clear to everyone? Is it clear? That's very good. Now let's move on to the metaphase. After this, which phase comes? Sir, metaphase comes. Very good. Metaphase, we are going to discuss about the metaphase here. So if I talk about the metaphase, what's the, like, if suppose you came to my house, so you will be able to know that, okay, Sir's house is maybe white in color. So you will be able to recognize that, okay, this is the Sir's house. And in the similar manner, how you're going to recognize the metaphase? So whenever you're going to see it under the microscope and you're going to see that there is no presence of nuclear membrane, then you can conclude that, okay, metaphase has been started. So again, this is divided into early and late. So let's write here. First of all, let's discuss the early thing. So in early metaphase, what happens? Nuclear membrane, I'm writing nuclear membrane as NM. So nuclear membrane gets what? Nuclear membrane gets disappeared. Nuclear membrane gets totally disappeared. No nuclear membrane is present. Okay. So I can say complete disappearance of nuclear membrane. Now what, sir? Now what? Now is that in this stage we can study the morphology of chromosomes. Morphology means what? How does chromosomes look? How many hands they have? How many chromatids they have? What is their shape? Everything can be studied in this metaphase. Morphology of chromosome. Best phase to study morphology of chromosome is what? Is the metaphase. Clear? Okay, so now next we have in the late prophase, what happens? Sorry, in the late metaphase, what happens? In the late metaphase, spindle fiber which was produced by the centrosomes, they come and they attach to the kinetochore and all the chromosomes gets aligned in an equatorial plate, which is also known as metaphasic plate. So let's see what is happening here in late prophase. So this was our chromosomes, right? Chromosome of S phase, and here we had what? Here we had the kinetochore. You remembered? Yes, sir. Yes, clear that. So this is the kinetochore, and here what is going to happen? The spindle fiber is going to attach and they're going to take these all chromosomes into a center position, which is known as metaphasic plate. Okay, so how it is going to happen? See, let's suppose that this is a cell. nuclear membrane disappeared. Okay, so in the late metaphase, the chromosomes is gonna look something like this. Let's have few of the chromosomes here. Okay, and then we will be having the spindle fibers. So I'll make it spindle fibers with white color. They're going to get attached with the kinetochore, and now the spindle fiber is gonna pull the chromosomes, and all the chromosomes will get arranged like this in the metaphasic plate. All of them will get arranged like this. So this is known as metaphasic plate or equatorial plate. And where it is found? It is found in the metaphase. Obviously, it is found during the metaphase. Now, what should we write here? We should write during the late metaphase, spindle fiber. Spindle fiber attaches where? It attaches to the kinetochore. It attaches to the kinetochore. And what happens? Chromosomes are moved towards the equator. And chromosomes are what? Chromosomes are moved towards equator. Towards equator means from the poles they are moved towards the equator. So can you clear what is poles and equator? Yes, definitely. So this structure, the top poles, just like our earth, so these are known as poles and this is known as equator. Clear? This is what happens in the metaphase. Now here comes the main thing. Here comes the main thing, which is anaphase. So in anaphase, what happens? So anaphase is the main thing of the mitosis. Okay, how, sir? See, first of all, I'm going to make you understand how this is going to divide. Okay, so till now we had chromosomes like this. So this was the one chromatid. Okay, I'll be little side. This was the one chromatid. Okay, and let's make the another chromatid with other color. This was the other chromatid. Okay, clear? Yes, sir, it's clear. This was N and this was N, and both of them was making 2N. Is it clear? Yes, it's also clear. Now what happens during the anaphase? During the before that, what was the DNA content here? The DNA content was what? Was it 2C, 4C? What? It was 2C. Yes, it was 2C. Okay. Now, in a similar manner, this is one chromosome, right? So in the similar manner, its homologous pair was also present, right? So let's make the homologous pair also. Homologous pair also. So we have this homologous pair. Clear? So here also we had what? I made a mistake. This is what? This is just the N. Okay. This is just the N. Okay. And this was also N. Now, the homologous pair is also present. So it is having how many content of DNA? This was also 2C, right? And that's what we were saying, that both of them made 4C during the S phase, right? And this was 2N. But now things is gonna change. Let's take one thing, okay? Let's take one chromosome here and let us understand what is gonna happen. So this is the one chromatid and this one is the other chromatids, okay? Oh, what I have made. This was the one chromatid and this was the other chromatid. So now what? Now will be, this was the N, okay? But now it will become 2N. How, sir? The chromatid is gonna break. The centromere is gonna break. So see here. Now we have both single chromatids. And now we can say that this is N and this is N. And this is how the chromatids in anaphase is going to divide. So now what is going to happen here? Let's come back here. This is going to divide like this, right? N, and this is going to be like N. So we have N and N, and in a similar manner its homologous pair is also going to divide. So this will be also what? This will be also like this, and this will be also like this, right? This will be N and N. So now in the cell we have total 4 and 4C condition. So in anaphase we can have 4 and 4C condition. How, sir? Let us understand this. Okay, let us understand this one more time, small step at a time, very easy concepts. Okay, now here we have during the anaphase, what is happening? Chromosomes are getting splitted. Okay, chromosomes are getting splitted. If you are joining my class right now, this class starts from 4 o'clock, and in this session we are going to focus on the droppers basically. And if you are a fresher student, you can join my class at 6:30. Now this in anaphase, what happens? In anaphase the chromosome splits. The chromosome splits and they form what? They form what? From the centromere. How chromosome is splitting? From the centromere, which we just now saw. This was the centromere, and from here only the chromosome is splitting, and that's how this N chromosome is now what? Is now made N and N. Clear? Great, sir. Okay, so chromosomes will split. Clear. Now they will start moving towards the pole where it was present in the metaphase. They were present like this in the equatorial plate, right? They were present in the equatorial plate, and in anaphase what happened? They broke. This was what? This was N, and now they broke from the centromere. So now we can say that this N is what? This N is basically, the whole was N. Now each chromatids can be considered as N. Remember in the starting of the class I said that when they are attached to a single chromatid, it's considered as N. When they are broken apart and now they have separate chromatids, each we will going to call it as N and N. Okay, so this will be now N and N. Clear? So this is how in anaphase they are going to divide. And here we have what? We have the spindle fibers. So spindle fiber is attached here and the spindle fiber is attached here also. So now the chromosomes will move. Now these chromosomes is gonna move like this. How they are gonna move, sir? See, this will be what? This will be the condition. They are going, this is the centromere, they are going to pull it like this. Okay, suppose this is the chromatids and they're going to pull it like this. Okay, so from the chromatid they are pulling, so the chromatids will be what? The leading end will be what? This top thing, right? So this will be called as leading and this will be called as trailing end. This will be called as trailing end. Clear to everyone? This part is what? This part is the centromere. So I can say that centromere is leading and chromatids is trailing end. So first centromere will reach to the poles and then and after that the chromatids will reach towards the pole, although they are going simultaneously. But among these two parts, the first one will be the centromere to reach the end. Okay, now anaphase is completed. Now let's get back to telophase. Now telophase is nothing. You don't need to study this telophase. Yes, seriously I am telling it. You don't need to study this telophase. Why, sir? Because this is just reverse of prophase. This is just reverse of prophase. Whatever we discussed in prophase, everything is gonna be reversed. Like how, sir? Like chromosomes will lose their identity. Remember, the condensation of chromosomes started in the prophase. Again it will be reversed. So now again we will be called then those chromosomes as chromatin fiber only. So I can write here chromosomes will lose their identity, And if I talk about what happens to nuclear membrane? Again, it is reformed. In prophase, nuclear membrane was out. Nuclear membrane was disintegrated. But here again, nuclear membrane will come. Nuclear membrane will appear. Right? And next, what is gonna happen? Cell organelles were also out. So in this case, what's gonna happen? Cell organelle will also be back. So I should write here, cell organelles are back. Cell organelles are back. And after this, when telophase is completed, what is going to happen? I'm gonna say that division of nucleus is completed, right? And then what's gonna happen? Cytokinesis. So after telophase, I can say karyokinesis is completed. Karyokinesis is completed. Now, how karyokinesis got completed? So let's get back to the anaphase here. Okay, let's get back to the anaphase. So during the anaphase, when the chromosomes were, like the chromatids were breaking, so the cells were doing what? The cell, basically this was the cell inside which the phenomena was happening. That cell formed a very little furrow, okay, and here in this nucleus was divided, and the starting of cytokinesis took place within this anaphase only, and in this anaphase only the karyokinesis also got completed, and in telophase it's completely completed. Okay, so starting of karyokinesis, it started in the anaphase only, it got completed in telophase, and cytokinesis also got completed in the telophase. So this is how the cell divides mitotically. Now, there are two things which we need to discuss, that cytokinesis in animal cell and karyokinesis and cytokinesis in plant cell. So sir, here we have a doubt, because in animal cells, sir, we have a cell membrane, a flexible cell membrane is present, but in plant cell we have what? We have a rigid cell wall. So how does the division is going to happen? So see here, in animal cell, what is going to happen? The things is going to be formed from outside to inside. There will be the arrangement of actin fibers. What is gonna come here? In the center, actin fibers is gonna come. Okay, actin fibers are gonna come, and then there will be the furrow formation. There will be the furrow formation, and finally this furrow will grow and the cell is gonna divide. This happens in the animal cell. But sir, what happens in the plant cell, sir? So in the plant cell, it can't be from outside, right? Because of the hard cell wall. So in plant cell, the division is from inside to outside. So how, sir? See, if this is a plant cell, so in the plant cell, there will be some deposition of structures. What structures, sir? What structure? The structures are known as phragmoplast. The structure is known as what? Phragmoplast. So there will be the formation of phragmoplast here. Okay, formation of phragmoplast. And how this structure is made? This structure is made by vesicles. This structure is made by vesicles, and these vesicles are formed by Golgi bodies. They are formed by Golgi bodies, and finally, like suppose the deposition started here, again it will be deposited here, again it will be deposited here, again here, here, and this is how it is gonna divide this cell into two parts or two cells. So I can say that in animal cell, the division is what? The division is centripetal. But in plant cell, the division is what? The division is centrifugal. Is it clear to everyone or not? In plant cell, the division is what? The division is centrifugal from inside to outside. And in animal cell, the division is what? The division is centripetal from outside to inside. I hope that this thing is pretty much clear to you all, right? Now let's move on. And let's talk about few other things, that what is the significance of mitosis, why do we need mitosis, and before that let's discuss about an important phenomena related to karyokinesis. So in karyokinesis, we are knowing that it's basically related to the division of the nucleus. Yes sir, we know that. So in karyokinesis, what if cytokinesis takes place, what if karyokinesis takes place but cytokinesis doesn't? Let's suppose the nucleus divided. In anaphase, the nucleus divided. Okay, we had this, but the cell doesn't wants to do cytokinesis. So we are going to have two nucleus but a single cell. Such type of condition is known as what? It is known as syncyticum. Such type of condition is known as what? Syncyticum. Whereas cytoplasm division, cytokinesis, is not followed by karyokinesis. When cytokinesis doesn't happen after karyokinesis, this is the condition known as syncyticum. Any example of this syncyticum? Anyone? Any example of this syncyticum? Anyone? So the example is what? The example is coconut endosperm. And this is what this comes in exams. It's a favorite question for NEET ones. So right here, give it a star mark here, okay? Give it a star mark. The coconut endosperm shows the example of syncyticum condition. Clear to everyone? Now let's have a significance of mitosis. So it's a very small topic. Like, why do we need mitosis, sir? The mitosis, why do we need, sir? If suppose I come to your home and someone came to your home and just taken a knife and cut like this. So to heal that cut, what is going to happen? Mitosis is going to happen. So can I say that for repair of cells, damaged cells, repair of damaged cells, mitosis takes place. Now what? They preserve the genetic material, sir. How, sir? How they are preserving the genetic material? How? Any option? Any idea? A very good statement, right? They preserve the genetic material. How? Because if I am 2n, then my daughter will be 2n, my son will be 2n. If a cell is 2n and is undergoing mitosis, then if suppose I am producing baby from mitosis, then my daughter will be 2n, my son will be 2n, right? If a bacteria is producing babies with the help of mitosis, 2n. So there is no change in DNA. There is no change, and there is not a single change in DNA content. So I can say that my DNA is preserved. So through mitosis, DNA is preserved. Clear? Now, what is happening here? Mitotic divisions. Now, in plants, where the mitotic divisions takes place? In plants, mitotic divisions takes place in meristematic tissue. Meristematic tissue is a place where the mitotic divisions takes place in the plants. So this was all about the mitosis. We have discussed a major part of the cell cycle. If you have missed the previous part of this session, in the previous part we have discussed very well about things like what is N, how N, what is 2C, what is 4C, how 4C came, why do we call that S phase contains the double content. Everything we have discussed in this particular video. So if you have missed any part of this particular video, watch it in 1.5x after your class. And do remember that we are going to have a quiz session of this particular chapter at 11 a.m. on Sunday. And if you are very new to this channel, subscribe to this channel so that you can get such awesome video. Till then, bye bye everyone.